Today we're going to be talking about enzyme kinetics. So essentially enzyme kinetics is the study of how enzymes move, or how they actually increase the rates of the reactions, okay? So like I said in the last video, this topic is very intense. It's very math heavy, okay? So you're actually going to be working with some formulas, right? And it's not fun, okay? So I'll be explaining it, but you should be paying attention, okay? So let's start with what exactly are we doing, okay? So enzymes, right? Enzyme kinetics. Okay, so what is that? So let's say that we have a reaction happening, okay? So we have substrate A is going to combine with substrate D, and they're going to give us a product that could be reversible, and let's call that P, okay? Now an enzyme is going to say, hey, you know, this reaction is taking too long. You know, I have a hot pocket in the microwave. I have to go eat that really quickly, so we're going to have to wrap this up really, really fast. So I'm going to help out, and we're going to do some kinetics, is what the enzyme said. So the enzyme, let's call that E, is going to be reacting with A and B. Let's say that there are multiple enzymes, okay? So what is the rate at which it changes? Well, whenever an enzyme reacts with the reaction, you're going to have some weird stuff. You're going to have that the concentration of A is going to change. Delta, which is a triangle, means a change over a period of time of t. So this means that the concentration of A is going to change per unit of time, okay? And it also means that the concentration of substrate B is going to change over time as well. And it also means that the concentration of the product, how much product we have, is going to change over time as well, okay? So you'll notice that for A and B, I have these little negative signs. Why do I have a negative sign? Well, whenever the enzyme is reacting with the substrate, it's going to be using up the amount of substrates available, right? So if I keep using, let's say I'm making a sandwich, okay? So the sandwich is a product, but I have the bread and I have the meat, right? So those are substrates. Well, if I keep on using bread, I'm gonna run out of bread eventually, right? And so the concentration of the bread, right? Let's say that this is bread, is going to go down over time because I'm using my supplies. And also the concentration of meat is also going to go down over time, right? But when I keep combining my ingredients, my substrates, let's say I'm the enzyme, okay, so I'm allowing this reaction to occur faster, right? Then the concentration of my sandwiches is going to go up over time, right? Personally, I like making Reubens. I think those are really good. So if I keep using my rye bread and my pastrami, they're going to keep going down. I'm going to run out eventually. But the amount of Reuben sandwiches that I have are going to increase. And so that's a really good reaction. We want to do that. So what I just described to you is called the velocity, okay? So this is velocity. Velocity, if you remember, is just distance over time, right? So actually it's displacement. Displacement, let's do that, okay? Over what? Time. Likewise, you can think of the velocity of the reaction as the disappearance of the substrate over time, okay? So this right here, what I just wrote, is velocity. Okay? So there we go. Now, velocity is not the product over time, okay? That is not velocity. Not velocity, okay? Because velocity is actually the substrate. So we're going to have the concentration of the substrate over what? Over some unit of time, right? So the change of time. It could be an hour, it could be a minute, a day, etc. So this right here is not velocity because this is not a substrate, that is a product, okay? So again, the velocity is actually the reaction rate. How fast does a reaction go? Does it take 10 minutes? 2 minutes? What is it? right? And essentially it's very dependent on the substrate concentration, okay? So velocity is dependent on, now whenever I have something in brackets, it's usually going to mean concentration, okay? On the concentration of the substrate, okay? This can also be shown as bracket S, okay, for future reference. So there you go. Now we're going to start getting into the mathematical portion of enzymes, and it's not my fault that this exists, right? It's actually a very well documented equation by two nerds, okay? So the name is called this right here, so I'm going to pronounce it for you too, right? And let's call that EQN, okay? So EQN is equation. So this is the Michelis-Mentens equation, okay? So Michelis was a German scientist, and Mentens was a Canadian scientist, okay? So the German and Canadian came together, and they wanted to describe enzyme reaction rates. They wanted to know how enzymes operated. So they looked at it, and they said, okay, well, obviously the substrate is going to go to the product, okay? This is trivial, right? If you're going to have a substrate, eventually some reaction is going to happen, and you're going to make a product. Yeah, whatever, who cares, right? But then they're like using their two brain cells, right? They're smart dudes, and they were like, okay, well, what if there are some steps between S and P, okay? So if I have my enzyme, and then I add a substrate that the enzyme can react with, okay, so they're compatible, we're eventually going to have ES, okay? Now you remember what ES was. Do you see an asterisk? No, you don't see an asterisk. So the ES is not, NOT, not the transition state, okay? This is not the transition state. It is actually the ES complex, okay? So you're just getting some Legos and you're connecting them together. They're not operating, they're just being connected, okay? The transition state, again, is when they start like powering on and they start doing reactions between them. But right now we're not doing reactions between them. We're just connecting them, okay? So this is the ES transition state. Or transition complex. Transition state, yeah. So here we go. And this could be reversible, right? Because we could go from the ES back to the reactants, or we can make a product. So on the top part, you can call this right here K1, okay? And this one could be K-1. So what does that mean? Well, K1 and K-1, those are just rate constants. So how fast does the formation of the ES, okay, complex form? And the K-1 is how fast does the ES degrade and go back into the reactants, okay? So how fast does that happen, okay? And it's safe to say that the ES is very stable. The amount of ES is very stable because there's an equal amount of it being formed and being broken, okay? So if you make 2 ES, you have another 2 being broken down to make the enzyme and the substrate, okay? So that's all they're saying. And they're like, okay, well, obviously we can make some product. We can make some product, right? We can make some product, and what are we going to call that? Okay, well, we can call that, I don't like that. We can call that, what? P, right? So we have the enzyme and the product. So we know that the enzyme is never used up during a reaction. It's just kind of recycled, right? And that's the product, okay? Now, of course, that itself is going to have a rate constant, okay? So that's K2. Right. Now, this equation makes 2 assumptions, okay? And the assumption is that, so you know how E and S can combine to make ES, right? And they can break down again to make the reactants. Well, notice that I do not have a backward arrow, okay? So this does not exist, okay? No, that's bad. No. Why? Well, we're making an assumption that the product is so favorable that we're always going to want to make the product. We're never going to want to make the ES, okay? So we go from ES, and 100% of the time we're going to make the product. Okay, so that is what is the the assumption. So P to S is never considered. So the fully reversed reaction is never considered, okay? And it also makes an assumption that, well, the reason why this never happens is because the concentration for P in the beginning is so low, right? It's never gonna go back to S. So what Michelis-Mentens was saying is, okay, well, the substrate is going to make the product, okay? But the product is never going to make the substrate because initially, at the very beginning, there's like 0.0000% product. And that's not enough product to go back to the substrate. And so therefore, the reverse reaction is not possible. It's always going to go from the substrate to the what? Product, okay? And like I said, the ES complex is essentially, it's a steady state environment. So it's not going to be unstable, I guess. But what essentially that means is that the concentration of the ES constant is constant because it's being produced and broken down at the same time, okay? So it's going to be very stable and it's going to form from the enzyme and the substrate, okay? So it's very stable. There are equal amounts of ES being formed and equal amounts of ES being broken down. Alright, so now we actually go into the mathematical equation, okay? So essentially, you have that V, right? So you have V, which is the velocity of the reaction rate, and that's going to be equal to Vmax, okay, multiplied by the concentration of the substrate that you have, right? And then you're going to divide that by Km. Now Km is called the Michelis constant, okay, and that's different for every enzyme that you have, okay, and I'll actually show you how to derive that for the Km. And then you're going to add the substrate, the concentration of the substrate again, okay? So here we go. So how do you get Vmax? Well, Vmax is kind of done experimentally, okay? Or you can solve for it if you have the other parts of the equation. But essentially Vmax is like a scientist goes into a lab and he wants to study the reaction rate for ATP synthase, okay? Well, he's going to put a lot of substrate, maybe like, I don't know, 50 grams of the substrate into the environment, and he's going to measure the maximum speed at which the reaction goes, okay? So essentially Vmax is just whenever the reaction is having a ton of substrates, okay? It's where the enzyme can be saturated. So Vmax, let's just say Vmax is done experimentally. It's usually given to you. Experimentally. So it's like tons of solute. Or I guess substrate. Right? Like the maximum velocity a reaction can go through. Okay, so there we go. Now Km is a little bit different. So Km, it's how much love the enzyme has for the substrate. Okay, so if you have an enzyme that doesn't want to be connected to the substrate, it's not going to be a very good reaction, right? So if you have a Km, well, I guess if you have an enzyme that has a lot of love for the substrate, you're going to have a good Km, okay? So mathematically, it's kind of like, it's a combination of the rate constant. So how long does it take for the enzyme to connect to the substrate? How short of a time does it take for the enzyme to bind with the substrate? Okay, so you could say that Km, okay, is a combination of the reaction rates for a specific enzyme. So, of course, carbonic anhydrase, which is an enzyme, is going to have a different Km than ATP synthase, okay? So those are just two examples. So it's a combination of the rate constant. So we can say something like, in the previous equation, you remember last page, you can have something like K2 plus K negative 1, and then divide that by K1, okay? Now the units are usually in concentration, so something like molar or whatever, okay? There's different units for it. And essentially, like I was saying, it's the measurement of affinity or binding, okay? So Km is actually the measurement of ES binding. Okay, so how much does the enzyme want to stick with the substrate? Okay, how much do they want to bind? Binding, okay? So there we go. And it's like the rate of the breakdown of ES, so how fast does this break down as an intermediate is the rate determining step of the reaction, okay? So essentially, K2 is usually the rate determining step. You remember K2 was between ES and the product. So how fast does the enzyme and the substrate break apart to make the product? If you find out how long that takes, that's usually the longest step, aka the rate determining step. Okay? So there we go. Hopefully that makes sense to you. Okay? And so what is a good Km? What is a bad Km? Well, over here, you can have kind of like a small Km, right, means that there is a tight, good bonding, okay? Means tight, good bonding. Okay, there we go. And what does a large Km mean? Well, a large Km means kind of like a large sloppy binding, large bad weak binding, I guess. Okay, so you can say that this is good and this is bad. Okay, so there we go. So essentially, it's kind of weird. But what does that mean? If we plug this idea into the equation, what does that mean? Well, if you have a large formation, so, let's just bring back the equation. Let's bring back the equation. If we have E plus S, can give you this right here. We have E plus P, and then we're going to have what? It was K1, K negative 1, and then we're going to have what? The K2. So if the forward reaction from E plus S into ES is super, super, super large, that's going to be really good, right? So imagine that K2 and K negative 1 add up to like 2, okay? But K1 was like 1,000,000. So 2 divided by 1,000,000 is a very small number. And if we have a very small Km, that's going to be really good. It means a tight, good bonding, okay? Right? However, if your K1 is very small, so ES, so E plus S makes ES very, very quickly, right? But then the K1, but the K negative 1 and the K2 are huge, that's going to give you a big number, right? So if K2 plus K negative 1 adds up to 10, but K1 is 1, well, 10, right? Km is equal to 10, and 10 is a big number. So that would make a large sloppy binding. And so essentially you want your K1, which is a forward reaction between the enzyme and the substrate that makes the ES, to be very, very, very large, right? So just think about that. Try to do it on your own free time, see the logic between it. But yeah, that's what it means for the Km, okay? Right, but we can also make another equation from this equation. So, you ever seen one of those Russian dolls where you open one up and there's a smaller one inside, and then you open one up again and there's a smaller one inside? It's kind of like this equation, but it's not fun. So, it's ridiculous, really. But remember how Km essentially has the same units as S. So we can say that Km has the same units as the concentration of S, right? So again, Km is just a concentration as well, so it's like molar or millimolar. Same thing for the substrate. So that could be like a molar of substrates or millimolar of substrates. Essentially, because they have the same the same concentration, we can do something weird. Well, what if we have a point of the reaction where the Km, right, is equal to the amount of substrate? So it's weird. It's like, what if we measured the enzyme's love for the substrate, but then that amount was equal to the amount of substrate available? Right? So if you like eating apple pie, and you can eat 2 apple pies in a sitting, and there happens to be 2 apple pies right in front of you, well, you're going to say, okay, well, my love for apple pie is 2, and I have 2 apple pies in front of me, so my love is equal to the amount available to me. What happens to the reaction then? How are you going to eat the apple pie? How fast are you going to eat it? Well, the Michelis-Mentens equation is going to describe it to us, okay? So we can say that in this weird example, we can say that the velocity at which you're going to eat the pies or do the reaction is going to be this. So Vmax, that's the maximum velocity possible, times the substrate, right? But let's say that you have Km is equal to Ks, okay? So if Km is equal to S, instead of writing Km, I could just put another S, right? That's mathematically allowed in here. And then we also have another S right here. Okay, well, S times another S, what is that? 2S, okay? That is 2S. And so I'm going to delete that and just write 2S, okay? And you know why I just put it there. So in math, we can actually cancel out like terms, okay? So we can cancel out this guy right here, right there. And we have derived another equation. We can say that the velocity is equal to the maximum velocity divided by 2, okay? So it's saying that if you have, it's saying that your love for pie in this example is 2, and there are 2 apple pies in front of you, well, how fast you're going to eat the pies is your maximum speed, let's say 15 minutes, divided by 2. So it's going to be what? Like 7.5 minutes each, right? So that's how this equation describes it. It says that the velocity of the reaction, whenever the love for the substrate is equal to the amount of substrate available, is going to be the maximum velocity of the reaction divided in 2, okay? That's all that is saying. Hopefully I have described it to you in a good enough example and it makes sense, okay? Because that is, a lot of people have issues with this equation. They don't know what it means, right? Okay, so there we go. It's pretty good. Let's go back to this top equation, okay? Let's go back to this equation. So let's say that Km is going to equal the amount of substrate, concentration of substrate, right? Well, what if there is a very small amount of substrate available? Let's say, I don't know, like 0.001, right? Let's just say. Well, now you have 0.001 times 0.001, right? And this substrate is also 0.001. Okay, but the maximum velocity, let's say, is 10 million. We're just pulling out random numbers, right? So 10 million times 0.001, I don't know. Okay, I really don't know. Let's say it's some number. Let's just call that a number, okay? Well, that number divided by a supremely tiny number is huge, right? If you divide 10 by 0.1, that's 100, right? So this huge number divided by a small number is a ginormous number, okay? And so if you get a ginormous number, you can plug it in a calculator, for an example, you're going to have a huge velocity, okay? And so we can say that a small km, let's do that right here, a small km can achieve a huge velocity. A huge velocity at a low concentration of S. At a low concentration of S. And of course, this is when Km is equal to S, right? So there you go. Sometimes it can just not even be equal to S. You just have to have a low enough concentration of S to have a huge velocity, okay? So I think that's pretty cool. And so in that same logic, if you have a high Km, let's kind of piggyback this idea, right? Let's piggyback off this idea. If you have a high, if you have a huge Km, we're going to need more substrates to achieve the maximum velocity, okay? Huge Km means more concentration of S is needed to achieve, and I really can't spell, the same velocity. Okay? More specifically, I'm talking about the half of Vmax, okay? That's what I mean. Now we're going to be talking about the turnover number. So what is a turnover number? Okay, well, the turnover number is actually represented as K cat. Now remember that K is going to be the rate constant. Well, cat just is shorthand for a catalyst, okay? So this is called the catalytic constant. Okay, now remember catalysis? Catalysis is where the enzyme just holds the substrate in a good position for it to react more efficiently. And so this is going to tell us how fast that occurs, okay? Okay, so it tells us, I'm going to write it over here. It tells us how fast the ES is formed to the enzyme plus the product, okay? And so that's like the catalytic cycle right here. So we have catalytic cycle, okay? So this is ENP right there. That's the catalytic cycle. So how many catalytic cycles does it do per unit of time, okay? This is a first rate order constant. So this is a first rate order constant. So it's only dependent on one variable, okay? Essentially what it is as follows right here, that the catalytic constant, okay, is equal to the maximum velocity divided by how many enzymes are present, okay? So this is the total enzyme concentration. Right, so that makes sense. Like, how fast can the reaction happen? And then we're going to take the maximum speed divided by how many enzymes are present, okay? And so we can tell the speed, okay, of how the enzyme and the substrate are going to combine to make the product. So that is just the kind of like the mathematical expression to find how fast the product is going to form. Okay, so I think that's pretty cool. Also, you know, like if we divide, we can make another one. We can also divide kcat divided by what? Km, and we can find the catalytic efficiency. Okay, so this is the catalytic efficiency. Of course, I misspelled it. Right, so that is the catalytic efficiency. Yeah. So how much does the enzyme grab the substrate and help it out, divided by how much does the enzyme love it? Okay. So how much does the enzyme love the substrate is going to be one of the main factors. Okay. So if you have a huge if you have a huge Km, right, that's going to be bad. You're not going to be very efficient, okay? But if you have a small Km, which is a tight bonding, you're going to be very efficient, right? The enzyme loves the substrate, so it's going to help out even more. And so the catalytic efficiency is going to be increased, okay? So there we go. All right, now we're going to be talking about the Lineweaver-Burk plot equation. So back in 1932, two scientists, Lineweaver and Burk, were looking at the Michelis-Mentens reaction, or equation, and they looked at it and they said, hey, you know, this kind of sucks. We can't really do anything because whenever we try to plot it, this is what we get, right? We get like some hyperbola, and that doesn't show us anything and doesn't tell us anything about the reaction. And so they looked at the equation. They looked at the Michelis-Mentens reaction, or equation. I don't know why I say reaction, but they saw this. They saw Vmax multiplied by the substrate over the Km plus the concentration of substrate, and all they did was they took the reciprocal. So they just turned everything upside down. So they have 1 over V is equal to what? It is equal to the Km over the Vmax times 1 over S plus 1 over Vmax. Okay, so of course they did some more stuff and they canceled out terms, but that is the end result, and that just comes from the reciprocal of the Michelis-Mentens reaction. Okay, so now this is a very important equation. So this is called the Lineweaver-Burk equation. And what it does is that it converts a hyperbola into a linear graph. Okay, converts to a linear graph. Now you're probably seeing something very familiar. Probably not. But look, this is y is equal to mx plus b. So isn't that kind of weird, right? So now you have this equation right here. We have that 1 over v, and we have km over vmax, and then we have 1 over s. Phenomenal. I love that. And then we also have 1 over vmax. Okay, so what does that equal to? Right? Well, this is equal to y, this is equal to m, and this is equal to x, and this is equal to b. Okay? And essentially, if you want to, if you have a plot, you can actually find the, the Km values, the Vmax value, the substrate values, right? Everything that you need, right? So I think that's pretty cool. So you can see from this picture right here, we have kind of like a graph, right? It's a linear graph. But notice that the slope, which is m, so this is going to be your m right here, their slope is actually the Km, right, divided by the Vmax. Okay, So the Michelis constant divided by the maximum velocity is your slope. The intercept for the y-axis is going to be your 1 over v max, right? Actually, not the well, yeah, so the intercept of the I never liked math. But essentially your intercept for the y portion, which is b, is going to be equal to 1 over v max, okay? So that's b. Alright, and then it says that essentially your intercept is negative 1 over km, which is kind of weird, but I think that's just how it is. Right here they're just talking about the 1 over s, right? So that's going to be your x. Now if you kind of know math a little bit, this would be your x-intercept, right? So this image is a bit more complicated, but essentially you just want to treat this as your x-intercept, right? Or your 1 over s. So let's recap. Your slope is just the Michelis constant divided by the maximum velocity. Your intercept is 1 over the maximum velocity. And then your x-intercept is just 1 over the substrate concentration, okay? So that's it. And you can actually find out a lot of things using the Lineweaver-Burk equation. So for this course, we're going to be using that equation a lot, especially when we're talking about inhibitors for enzymes. And yeah, let's try to do an example of how this can help us out. So let's say that on an exam or something, they want you to calculate the Vmax and the Km, and they gave you this weird thingy, right? They're like, well, here's a bunch of numbers, go for it. Alright, all you have to do is just convert the equation into the Lineweaver-Burk equation, okay? So we have that 1 over V is equal to what? What's it equal to? It's equal to the Km, or the Vmax, Vmax times 1 over S plus 1 over what? Vmax. Okay? That's all there is. So over here we can make a kind of like a table, right? We can say that y, and then 7 times 10 to the negative 8. And we can have x and 0.0071. So what are they equal to? Well, y is equal to 1 over v. 7 times 10 to the negative 8 is equal to km over Vmax. X is equal to 1 over s. And this number right here is equal to 1 over Vmax. Now you can see what I'm going to do, right? So I'm trying to figure out what Vmax is and what Km equals. Okay, well this is pretty easy. Over here they gave me my values already. So I can just kind of shift this guy over here, right? So if I do that, let's make this a bit better. It's an old trick. You can have 1 over 0.0071 is equal to your Vmax. Okay? And so we know that our Vmax is equal to 140, I want to say. Yeah, 140, okay? So there we go. So we have that Vmax is equal to 140. Now this becomes a bit more simple. So we go back to this equation right here, right? We go back there. And we say, well, 7 times 10 to the negative 8 is equal to my Km value, okay, divided by what? My Vmax. But we know what Vmax is. We know that it's 140. So this becomes really, really simple. We have 7 times 10 to the negative 8 multiplied by 140, which is equal to your Km value. And we should get 1 times 10 to the negative 5. And remember the unit for Km is concentration, so let's say molar. Okay, so essentially that's how you calculate the Vmax and the Km. Okay, so there you go. Sometimes you have to solve for one or the other, so it'll be like solve for V and S, and they give you like Km and Vmax, right? So that's all you really need to do. Hopefully that helps you out, and now you know how to use the Lineweaver-Burk equation. Alright, so thank you so much for watching this video and for spending your time with me. I really appreciate it, and I hope that you do well on your exams. Right? So thank you and have a great day. I love you.